focus more on the clinical aspects of APDT. You've heard a lot today about the mechanism of action. We recognized early on that the barriers to entry to clinical application were probably more along the lines of some of the things that Mark has just mentioned. One, the access to capital. Two, access to regulatory approvals. You'll see that over the course of the last decade, $18 million has been spent on developing this technique using only methylene blue. So I have a great deal of respect and admiration for the people in this room and others developing complex photosensitizers, derivatives of sensitizers, different applications. We focus strictly on topical antimicrobial therapy for a variety of indications. I'll cover three of the largest ones for us. And using just methylene blue, we do have a variety of programs, one of which I'll touch on going forward, on developments associated with methylene blue, but the regulatory problems that you face are so significant that we're very careful about moving beyond the well-known substance. So, with apologies for the hyperbole, I'll just say that during the course of this presentation, 14 people in the US and the EU will die from an avoidable hospital-acquired infection. The key word there is avoidable. And from the O'Neill paper, which was published in 2016, you'll see that antimicrobial resistance will kill more people by 2050 than cancer and diabetes combined. So we're running into a very significant problem. Mark touched on it. With thanks to Mike Wilson for this slide, it was recognized very early on that one of the issues with antibiotics, antimicrobials in general, is that resistance follows them like a faithful shadow. Why is it being upregulated? Primarily because of the massive antibiotic misuse in animal feed, where it's used as a growth promoter. Very difficult to avoid this in the food chain, whether you're actually a vegetarian or pescatarian or you eat meat. The latest surveillance data has just come out for 2016 for the European antimicrobial resistance surveillance, which occurs every few years. And here you see on the right the level of isolates completely resistant to 5 major antimicrobial groups. This is for E. coli, similar for Staphylococci and other gram-positives. But in this gram-negative, which is ubiquitous, you see how approximately half of the isolates from a variety of countries are still susceptible with a single potent antimicrobial, but they're starting to win, and this green line gets smaller and smaller every single year. And that's really one of the most important reasons for this extraordinary cost in healthcare systems of both the US, this is US numbers only, as well as elsewhere in the world. It turns out that in Great Britain you have a very good control of staphylococcal species, but it's beginning to creep up here as well. It's unavoidable because of international travel causing gene modification through what's called the environmental resistome. But this is also a reason why, Mark touched on this, it's so difficult to get a handle or get on top of the resistance factor. It costs so much money and it takes so long to develop a single antibiotic. And of course, when that antibiotic is released to manufacturing, resistance will occur in as little as 6 months. And so we're probably going to see a black box which says only to be used for the worst complicated skin structure infections or deep space organ infections, which limits revenue. So this number is disputed. It involves some opportunity cost as well, but somewhere between $1 and $2 billion to develop a new antibiotic and take it all the way through, with the result that few antibiotics are being developed, and we have a major problem. Our company started 15 years ago on specifically this aspect to approach the problem of antimicrobial resistance from a very different perspective, leveraging some of the work that has happened over the last 100 years in methylene blue. We focus entirely on this drug, the cationic nature causing some selectivity to anionic, both gram-negatives and gram-positives. We pump at 664. We almost always focus on 150 milliwatts per centimeter squared and about 10 to 50 joules, about 1/5 of that or 1/10 of that in joules per square centimeter fluence. And we keep everything about the same. We're really about how we apply this clinically, and I'll go through a lot of what we've done. It's taken about $80 million, as I said, to get here. And that's just an indication of how expensive it is, even with a simple antimicrobial. We have a variety of products in the pipeline, and I'll talk about the top 3 and why they're so significant, we think, to human healthcare going forward. Some of which have started to be sold, but sales are early for us, primarily in Canada. And we've just received some European approvals, which I think you're going to start to see a little bit more of us in the future. I'm going to just spend a quick second on this slide. It takes a long time, and it takes a great deal of outside the lab thinking to drive adoption of a new approach, much less a new drug. Your protocols have to be improved. You have to worry about NGOs and government advocacy. You have to really worry about reimbursement. It doesn't matter how inexpensive you build something, unless it's reimbursed, it won't be adopted. And the thought leaders all have to be behind you to avoid both, so we say, subtle and less than subtle cannibalization and issues with what is said about the product and indeed the entire approach to get to where you want to be, which is a paradigm shift. And this can take 5 to 10 years. And it's really one of the more difficult things to accomplish as a company, because one starts in the lab, you get great results in a petri dish, and then to migrate that and translate that into a functional product takes a great deal of time and money. In particular, with methylene blue, and that was a great question from the audience just a little while ago about why these dyes might not have been approved in the past. The fact that you can buy this as a drug for injection, primarily as a hemoglobinemia reversal agent, doesn't mean that you can adopt it into, for example, a new indication like APET. You have to go through the development almost as though it were a new chemical entity, starting from scratch with making sure that the regulators like exactly what you're doing, residual solvents, purity levels have to be assayed in the almost 100% range. Your API has to be kept extremely low of contaminants, and indeed, because of those contaminants and heavy levels in particular, most dyes which would be used for the textile industry were toxic and genotoxic to boot, so a not particularly good basis to start with a new dye technology. So what are the photodynamic parameters? I think you may have heard this from some of the other speakers. Power is important. If you get too much laser power to activate your dye, your thermal effects will start to predominate, start to create tissue necrosis. Be very careful with the regulators in this space. Clinical relevance requires you to be able to deploy something rapidly. If it takes a week or 10 days to do something, there are alternates in the, should we say, normal antimicrobial space or conventional antimicrobial space that will compete with you. And so one of the advantages of APET as a modality is the fact that it can be rather fast. You also have to be pretty careful about this kill, because the therapeutic window will limit the applicability if you're starting to kill too many human cells. So it's actually quite a trick to get just enough photodynamic parameters to get what you want, which is a killing of the surface microbes, often in a biofilm form, and avoid killing of human tissue. We also do quite a lot of work in the chemical domain, and I'll just show this one slide. You'll see, for example, that we conventionally test products in the dark with no light, and we see here that 10 to the 6 to 10 to the 7 CFU per mil of these microbes, in this particular case MRSA, remain. And then if we apply methylene blue, it is very slightly microbicidal, and that has been shown in the absence of light over many years. But with the presence of light, we can get around a 3-log kill. Well, we work with powerful amplifiers that themselves, as you see, do not contribute any further killing directly, but in the presence of light can take the 3-log kill of methylene blue and completely eliminate any microbes remaining. And that's only in 30 seconds. So these are 60 and 30-second kills. So in 30 seconds, we can remove 1 million colony-forming units of MRSA, which no other antimicrobial has ever shown the capability of doing. And we can do so safely in contact with human tissue. That's the real, I think, allure of this technique. There are very few of us working in the area, but those of us who are doing so are seeing results which are really extraordinary. You're seeing some recent entries now into the commercial space, among them a company based in Sussex called Destiny Pharma, which just did a small IPO in London, to use this, albeit with a compound while being a sensitizer, is no longer being activated by light because of the high levels of dark toxicity that they actually do have. This is the kind of therapeutic window showing how in the absence of light absolutely nothing occurs, and so the patient can be bathed in the substance without any negative effects. Importantly, when bugs grow, they do so in biofilm. That's a large amount of proteoglycans and other thick cross-linked material which inhibits access to the bacteria themselves, and these cationic stains can quickly penetrate these 300 micron thick films and eliminate them. Importantly, again, non-resistance forming, we heard about this because of the speed of action, the methods of action, the site of action. We've taken reculturing experiments out for 2 years and we still can't show any diminution in mean inhibitory concentration required to completely eliminate these final populations of microbes. They die just as quickly as 2 years earlier after multiple reculturing experiments. So we've sort of proven the fact that it's non-resistance forming. I refer you to the work of Mike Hamblin and Tanaka et al. At Harvard-Wellman to show the other elements here of APDT, specifically using methylene blue, where there is some impact on local adaptive immunity, and the system composed of organism, vasculature, and primarily neutrophils will create an adaptive immune response. And so if you notice here, the photosensitizer and light have been applied 1 day prior to infection, and it is only in the irradiated animals that control of the microorganism can occur over a period of time versus the untreated animals, which do not show that effect. So prior administration of PDT, specifically using methylene blue, can result in an adaptive immunity, and that's an interesting phenomenon because it means that once treated, the area is difficult to reinfect. That's been seen in prior work in herpes simplex. This is the first time it's been showed in bacteria. This is an MRSA murine knee arthritis model. It's a fascinating paper. So just to summarize, the topical APDT aspect does destroy biofilm, and that's what we talk about primarily, but there are many other interactions with the human host, including the destruction of host-derived and bug-derived cytokines, proteases, chemokines, and so forth. There's a downregulation of the inflammatory response through this adaptive immunity sequence, and so the combination of a potent antimicrobial with a potent anti-inflammatory produces this improved outcome. And we could go on for days discussing that. So I'm just going to cover some of the outcomes and applications of this technique that we have taken to the market. Debridement therapy does not cause the problem to resolve. We find these lesions, place the drug into the lesions, activate by light. 664 nanometer light at 150 milliwatts per centimeter squared, takes 60 seconds. And then the drug is formulated with adjuvant matrices and excipients to stop any staining. And you get these great outcomes. If you were a dental practitioner, you'd see these 9 millimeter pockets, essentially centimeter deep lesions in the bone and tissue above the teeth coming down to 3s and 2s, which are essentially the way you were when you were born. So at the bone level, you can see fundamental changes. This is probably why the technique is gaining a lot of traction in countries where this is approved. And this system is available now in various countries, though not yet in the US. At the bone level, these teeth would have come out of this patient's head. This is a young 12-year-old, but you can see how with administration of the photosensitizer and light, the elimination of the microbes results in regrowth of the bone, which otherwise would have required explantation of the tooth and an implant. And you can see the effects on blood in various different parameters, including C-reactive protein. The system that really is beginning to develop a lot of traction now is a system called MRSA, which is designed specifically to remove the colonization of microbes from your nose. 40% of adults carry Staph aureus in the nose, and there's a high mortality rate, about the same as Ebola if it gets into your bloodstream after an infection, after surgical incision. It's being demonstrated that if you simply remove it from the nose, especially in the resistant forms, you can really significantly reduce hospital-acquired infections, specifically related to surgery. But the drug that is currently used, mupirocin, is starting to show very high levels of resistance here. So this is a real patient used with permission at the largest acute care hospital in Canada. You see a little swab with the drug that is being applied to the nose, and then the light radiator is brought into the nose. These are disposable radiators. Just go back a couple slides, you see how we break open this popule with the pre-saturated swab, and then the light radiators at the tip. And these are all made in gigantic production processes, so that can be made inexpensively in the hospital. Really doesn't have to put out too much money to avoid the pharmacoeconomic barrier. This takes around 60 seconds, up to 4 minutes, depending on the protocol being used. It can be done at any time. Imagine the workflow advantage of a 2-minute therapy, which can reduce hospital-acquired infection rates by 60%. The patient can go in for surgery and have it right as they're lying on the gurney being rolled in. It can happen 4 hours before. It's a really significant evolution, despite very simple photodynamics. And from actual swabs with real patients, you can see how we can come down there's a chromaga swab that's a plate that's specific to MRSA. These patients come in really contaminated. A patient like this on the left is going to be contaminating the whole hospital. The healthcare workers, the surfaces themselves, their own wounds, and in many cases, I would say the majority of cases, the patients are not even swabbed to determine if they're carriers. And that's one of the reasons we have a $45 billion problem today. I'm just going to run you quickly through a large quality improvement project that was done at Vancouver General, which is Canada's largest acute care hospital. And in this paper that was published, the first paper involving a large deployment of APDT in the general population, ultimately, just taken as an end result, around 80% of the patients decolonized to 0 in those 4 minutes. And there's a non-significant difference between MRSA and MSSA species. In addition, not shown here, you're decolonizing other potentially pathogenic microorganisms. Another big advantage of APDT, because drugs like mupirocin are very narrow spectrum anti-staphylococcals. So this is taking care of Pseudomonas, Klebsiella, H. flu, all of the various hospital pathogens that the patient would otherwise breathe upon their own wounds. And again, this is a single dose. The effects last up to about a week, depending on what the patient is doing. No significant adverse events in the entire study, very well tolerated. And it's so quick that you're getting, it's also got a high-tech feel. There's that laser involved, and the patient's glowing for a while. You're being electronically disinfected. And you'd be amazed how rapidly it gets compliant with the nurses and with the patients. Well, that's the result. The surgical site infection rate, not the microbes that were reduced, but the actual rate of surgical site infections, down 78% over 4 years. And now in 2017, this has remained stable at just around a half of 1 infection per 100 patients. So seriously high impact. Strongly recommend that if you ever needed an invasive thoracotomy or some other procedure, that you get your nose electronically photodisinfected. Fourfold reduction in infection risk. This is in 35,000 patients. There must be close to 50,000 patients at this point through. And an annual savings of almost $2 million, which makes a big difference to a center like that. Either primary, secondary, or tertiary care facilities in developed nations today can really appreciate these kinds of savings. So simple radiators, although a lot of technology goes into that diffuser to make it inexpensive. No screening of the patient, no more PCR to determine if the patient's a carrier, no delay to the operating room while you wait for the mupirocin and the other antibiotics to work, no contact precautions to the staff in the hospital, and of course, in general, good stewardship because we are not increasing the mean inhibitory concentrations of antibiotics by overusing them. So really direct hit on primary antibiotic issues today. Let's go over a couple of the other products in a little less detail. Sine wave is a technique to disinfect the sinus cavities of patients who have resistant to any other therapy. Same thing, a methylene blue-based sensitizer with a light distributor, much bigger laser because these cavities are larger. And here you see real patients receiving ethmoid, maxillary, and in a cadaver, the frontal sinus being illuminated with the activation light. Outcomes dramatically outperform standard of care in chronic sinusitis. We believe this is going to be a great product for these patients. That's the sinonasal outcome test. It's a real quality of life questionnaire in sinus patients. And finally, in ventilated pneumonia, these tubes get colonized with large numbers of multi-species biofilm-forming organisms, and they drip into the lung. It might cost $2 for that tube, but it's a $3,700 average infection cost per that tube if you look at all the costs of empirical antibiotic coverage and other issues. Much longer light radius, much larger lasers, 3 log kills, and in general, we think that's going to be an important way to reduce ventilator-associated pneumonia in the clinic. So, in conclusion, simple physics, applicable to a wide range of disease, very rapid, very thorough disinfection, reduces antibiotic use, safe, and really extraordinary outcomes when you look at interventions that can save a hospital that kind of money and reduce patient risk to that degree. So obviously I had to rush through a decade of development, but thank you very much for it.